Katja Simon is in the immunology department here at Oxford University, and she's set up a line of inquiry concerning the investigation of autophagy in determining cell fate in the hepatoimmune system. And her group discovered that the main conserved cellular bulk degradation pathway can maintain healthy red cells, stem cells, and memory T cells, and can promote differentiation while preventing aging of the haematopoietic system. Now, we are therefore yet again honored by one of the most fundamental biological science contributors to the study of aging. And without any more ado, I'll ask Katja to give her presentation.
Thank you, Dennis. That's a kind introduction, a little bit over the top, but I will start again. I'm going to talk about the immune system and autophagy. So this leads on quite nicely from Tamotsu's talk, which was very interesting. I'm going to share my slide now with you. Okay, so I work at the Kennedy Institute of Rheumatology at Oxford University, and over the last 15 years, I've concentrated on the role of autophagy in the immune system. And overall, autophagy plays a role in the health of all of our cells. It maintains a healthy genome. It maintains a healthy proteome. So that means the proteins are not healthy, maintains healthy organelles, and also reduces inflammation. And I'm going to tell you a little bit how it does that in the immune system. So just a brief introduction to the autophagy pathway that Tamotsu introduced very nicely but I think a recap of that can't do any harm. So it's the formation of the double membrane vesicle, which is a new organelle in the cell, which can engulf cargo that's no longer needed in the cell. And it has got kind of several cues to when this happens. It happens when the cell starves, and that's been very well described, of course, by the Japanese groups. In response to starvation, the material that's engulfed and then degraded can be recycled and reused by the cell. But other cues can be cellular differentiation, stress, and so forth. So what happens is that this double membrane vesicle elongates and forms the autophagosome. And in this autophagosome, bulk material can be engulfed, such as whole organelles, protein aggregates, and so forth. And that then fuses with the lysosome, as said before, and building blocks can be reused by the cell, such as amino acids, free fatty acids. This, of course, has a huge effect on metabolism. And this is by summary, it recycles nutrients and it disposes of old organelles and unwanted bulk material. So you can imagine that in aging, this plays a major role where these accumulate. And this is in addition to another main degradation pathway in the cell, which is the proteasomal degradation pathway. And that is known to degrade single proteins or peptides. So this cannot engulf big materials such as these shown here. So I'm going to briefly introduce you to the immune system and what autophagy does in the immune system. The immune system, immune cells come from the haematopoietic stem cells, which is located in our bone marrow. And very, very simply put, there's two bits to the immune system. We've all become a really good immunologist over the last year. There is neutrophil and macrophages, which is the first line of defense. It's a short-lived response to any incoming invaders. And these cells also mediate inflammation. But there's also the adaptive immune response, which is based on T and B cells. This is the long-term immune response. These are really important in vaccination, but because they can remember, they're still around, years later after you've encountered your vaccine or your pathogens. So this is these cells we think are possibly more prone to aging because they're long-lived. But we also know that these cells age, even though they're short-lived, their progenitors will be long-lived. So our contribution to the roles of autophagy during this immune cell or blood cell differentiation is manifold. Over the last 15 years, I said I've been working on this, and basically these two parts of the immune system both have a problem when there's no autophagy in the cell. So most of this work is based on knocking out a key autophagy gene, ATG7 or 16 or 5. And I won't go into detail, but I just want to make it, so this is again the innate and the adaptive immune response. I just want to make it very briefly summarizing the main findings, which is that it's either autophagy is really important in differentiation of these cells. So if, for example, you knock out an ATG gene in the neutrophil, they won't even make it to the final stage of neutrophils, but will be arrested here when they don't have the function they need to have in the immune system. And the other major theme, if what autophagy helps with, is maintenance. And this, as I said earlier, the memory T cells and B cells are actually long-lived, and autophagy helps them to be maintained in a healthy way. And this is also true for the haematopoietic stem cell. And lastly, what we found in macrophages is that, and this is also true for other cell types, that autophagy prevents aging. And of course, this is the topic of today. So the three themes I like to introduce here is autophagy maintains a long life of long-lived cells. It helps differentiation of immune cells and prevents aging. So the major societal problem of the aging immune system, and we've already heard from this morning what an economic fallout that has, but here are the hallmarks of increased number, of what's happening with age. So there's an increased number of macrophages, but these don't work very well. And these usually happens quite dramatically after the age of 60, 65. And then at 80, here there's the curves I'm showing you, of course, are not curves that I drew, but at the age of 80, we'll probably all have these problems, whereas at 60, some of us will already and some won't have these problems. B cells go down with age in number, and so do T cells. These are both naive T cells and memory T and B cells. And then meanwhile, inflammageing, the excessive cytokine production increases. And overall, that leaves the organism susceptible to infections such as SARS-CoV-2, poor responses to vaccines and malignancies. And levels of autophagy over the course of the age of this lifespan fall. We don't know exactly when they start to fall. So this is a dotted line, and we're currently looking at that in the immune system. So signs of an aged immune system, these signs lead to being infectious disease, one of the fourth cause of death in the elderly. This is maybe an old slide, and that might be a bit higher now. We look at COVID, the reactivation of latent viruses. These are viruses that live with you. For example, shingles is caused by the chickenpox virus. We have a major complication for the elderly in hospital, which are the antibiotic resistant strains. And that's because the immune system doesn't work anymore. A lot of your microbiota in your gut is also changed. And there's a low-grade inflammation, which leads to the three other causes of death: heart disease and stroke and neurodegeneration and other related diseases such as Alzheimer's and Parkinson's. So this low-grade inflammation doesn't lead to it, but it contributes at least to these diseases. So understanding how the immune system ages and what we can do about it is really important. Okay, so in SARS-CoV-2, there've been many studies now showing that actually in the youthful system, there is a good response to the virus with not too much excessive innate immunity and enough adaptive immunity to clear the virus, whereas in the age system, the innate response takes over, and people die of their cytokine storm. So this could be, and this has been shown now, probably could be due to inflammageing and senescence, and in particular immune senescence, and this leads to the cytokine storm. So we're understanding more and more how aging also contributes to the failure to control viruses. So autophagy is reused in immune cells from blood. I told you about this. This was an early study where we bled some PhD students and some older professors, and we found that autophagy levels were older. This is really quite early on when we didn't know really much about autophagy in the aged immune system. So we took blood from these people and then found that the autophagy levels fell. And these were not starved themselves, but these are cells that were starved. We couldn't really find so much difference here because starvation of primary cells is really quite difficult, it turns out. After many years of work, we couldn't really do that. But another really quite good causal evidence that levels of autophagy falling with age leads to more aging is when you knock out autophagy, the overall haematopoietic system comes up with quite a typical aged features. These are the stem cells that fail to survive and reduce ability to generate the blood system. We find anemia. We find the accumulation of macrophages, as I told you earlier, the increased cytokines and inflammageing, and also reduced numbers of T and B cells. So it's pretty obvious that autophagy prevents aging of the immune system. So let me just take you through the next bit of data now, for which I have to explain how the immune system works. So when a virus enters our body, there's first an innate immune response, but when the adaptive immune response kicks in, and this takes about a week or so, then the cells that are responsive to the virus will be expanded, and then eventually all the ones that are not needed will die. But this expansion can be measured by how many cells we have, and I will use this in the subsequent slides. This then will be, and as the virus is under control, this will contract down to a memory population, which will just keep the very best cells for later encounters of the virus when it comes again. And this is also what vaccination is based on. We keep a small proportion of cells that respond to this virus. And meanwhile, the antibodies made by B cells, which are really important to keep the virus under control, that takes a little bit longer to come. So your peak T cells at CD4 and CD8 T cells, I won't go into detail what these are on day 8. And then your peak antibody response is basically on day 30. And then if you have been vaccinated, this takes a while, and that's why you can't go out and go crazy once you've been vaccinated. You need to wait until this immune response is really set up. And then these antibodies are made by B cells, but B cells too leave some cells behind. Sorry, the antibodies are made by B cells, but the B cells themselves are also kept as a memory population. So even if your antibodies have waned, you have a memory B cell response. I get a lot of these questions now. I can't find any antibodies to COVID-19. Does that mean I haven't had it, and did the vaccination not work? But that's because antibodies are usually not kept for so long in the body. They have a finite half-life, but the cells that make them will be there. Okay, so if you knock out autophagy in T cells, you find that even though in the normal response in a wild type mouse is typically, as I said, it starts to be very good at day between 7 and 14. And if you vaccinate again, it's faster and stronger. And that's the idea behind these 2 shots of vaccine. And also, of course, behind the idea that if you have a memory response, it's fast and you won't even notice that you'll encounter the virus again. But without autophagy, you get a primary good response, but this secondary response, this memory response, is very poor. So somehow memory T cells are not doing well without autophagy. And this looks very much like an old mouse. This is 24 months old mice. The secondary response is very poor, that memory response. So are these related? Is this because autophagy levels are low that we get this poor memory response? Well, the way to do that was to reintroduce autophagy. And we looked at a paper at the time by Frank Madeo, which showed that Spermidine could do this. Spermidine is a polyamine, very simple molecule. It's an endogenous metabolite. You can give it in the diet, but our cells make it ourselves. It's synthesized from arginine, and it's found in natto fermented soya beans. I could see the Japanese are smiling here. This is what they eat a lot, and it's very good for you. I mean, it's not very palatable, but I think when you're Japanese, it's okay. And that delays aging in yeast, worms, and mice when you give it in the diet. So we gave the old mice some spermidine to see if we could improve on this poor memory response. And by our amazing, it was amazing, the result. You don't get that very often in your career. And spermidine has really boosted this blue versus brown here in memory response, and it's also even improved the effector response. And when we gave it to mice that had no ATG7, so no autophagy in the T cells, the number of flu-specific killer T cells was actually no longer improved, which means that it at least doesn't work via another pathway and possibly via autophagy. So when we saw those data, we wanted to see whether that was also true in B cells. And again, in old mice, B cells, the memory response is very poor. Here you see it takes a bit longer to make these antibodies, as I told you. And upon secondary immunization, your anti-response will go up very fast and eliminate the virus quickly. In old people, in old mice, and the same is true for old people, this is not the case. And that looks very much like a knockout again. So if you knock out ATG7 in B cells, you have a similar picture. This was actually not a flu vaccine. This is a system model antigen. This is a model system where it's easier to measure these antibodies and the response to it, but it's very much the same to any vaccination or viral infection. So we then ask again, could we improve this with spermidine? And we found that in the same system, giving spermidine for 6 weeks to 24 months old mice, we could improve this, not quite to the level of what we'd seen in T cells, but definitely improve it over the old, not quite back to young levels. So we then spent the next 5 years or so to understand how spermidine improves autophagy and these responses. So we found, and I won't take you through the data, a pathway which shows that spermidine helps a translation factor called EIF5A to translate proteins, and this is a ribosome here, which brings in the amino acids to translate the messenger RNA. And these proteins that this translation factor helps are usually proline-rich proteins, particularly when they're prolines, 3 prolines in a row, which are very, very bulky residues. And so without this translection factor, this can't work. One of these proteins is a transcription factor called TFEB, and this transcription factor transcribes now genes, and this leads to, and these are genes that are involved in the autophagy pathway, and that helps the T and B cells to make it. So I won't bore you with the data of this, because I know there's also a lot of non-molecular biologists in the audience, but I want to say that we found that all of these are reduced with age in mice, in B cells. And then we went on to look in humans. So this was all done as a mouse experiment. And if we bleed people across this age range, you can see that TFEB levels fall quite dramatically after the age of 60, 65. And same is true for this hypusinated EI5A, which is the translation factor, but not quite so consistently, I would say. So this consistent loss of TFEB in peripheral blood monocytes from old human donors is quite striking. So can we improve these levels, and would that bring back responses in the old? So firstly, we did some kind of proxy experiments, because it's not always easy to do experiments in humans. We found that if we look at autophagy levels here on this axis, if we activate a T cell in a dish from a human person, we can see that autophagy levels are highest on around day 3, day 4. But if we take out spermidine endogenous, so internal, that the cell makes themselves, the spermidine, by a drug called DFO, we can see that the autophagy levels are completely abrogated. So spermidine is necessary in human T cells to see these autophagy levels. And if we then give back spermidine back, we can see that it improves these autophagy levels again. So this shows that the spermidine pathway in the cell is very important in T cells for their autophagy levels. Then we wanted to know whether autophagy levels actually corresponded to vaccine levels. And we used a hepatitis C virus vaccine. There was already existing samples in the freezer. This is by the Oxford Vaccine Group that made the Oxford AstraZeneca vaccine with the same platform of chimpanzee and chimpanzee adenovirus. So we primed, we looked at these samples to look at when autophagy was highest. And I won't go through details, but I just want to say that the T cells that were responding to the hepatitis C vaccine were mostly had highest level of autophagy. And in another vaccine trial, again, using the same some other adenovirus platform, we found, and this is to respiratory syncytial virus, it was found that the old the people made a much worse immune response. So we wanted to know if this much worse immune response, and this is measuring interferon gamma, so T cell responses, was down to autophagy levels. So, and could we improve these by giving them spermidine? So I'll just finish on that to show you that actually in this RSV trial, the old people made a worse immune response, and the an autophagic flux, and that the interferon gamma responses correlated nicely with how much autophagy these people had in their blood. To the last slide here, we found that giving spermidine to old T cells improve their autophagic flux and two markers of their function, which is perforin and interferon gamma. So we think altogether that spermidine can reintroduce autophagy to human T cells, and it can improve their function. So meanwhile, we're now looking at doing a clinical trial to see if spermidine can improve COVID vaccination in the elderly. This is done in Austria, because here we've already vaccinated the elderly in the UK. And we're also going to see if it improves flu vaccine. And another arm of the trial will look at to see whether it improves the memory response to COVID vaccine. So this is ongoing, and I can't tell you anything about that. So basically, conclusion is that we want to develop spermidine as a nutraceutical in doing vaccination, but we also would like to see if we can develop other drugs along this novel pathway we found, understand why autophagy and how autophagy is reduced with age. And this will have an impact on immune aging, on inflammation that leads to age-related diseases, and vaccines for the elderly. And here's just a slide to thank everyone in the lab. And some of these on my team are actually listening today. Mostly Garda did the human work, and Hanlin did the mouse work, and all my funders. Thank you.
Well, thank you very much indeed, Katja. Yet again, we've heard from people working at the very front line of the processes by which it might be possible to reverse aging, and particularly to boost the process by which dead cells or dying cells or useless cells are being removed. Before I come on to question direct to Professor Simon, let me just emphasize the Q&A system is working extremely well. I've noticed that our previous speakers have been meticulously answering questions put up on the Q&A that I did not manage to put in open discussion. So do please use the Q&A if you've got specific questions for any of the speakers, and they will get answered. I got an opening question, and then we've also got one on the Q&A that we want to look at. You've effectively shown the slowing of aging of the immune system. What interests me as an integrative physiologist is what is it that tells the system which cells to eliminate? I mean, what are the candidate cells for autophagy and why? Do we really understand that, or is this a vague area? How specific can we be, Katja?
So are you talking about apoptosis?
Yes. Well, I suppose that's what you would call it, a nice scientific term for it. Yes, we call it cell dying.
So basically, we accumulate senescent cells with age, and we think that if we can eliminate senescent cells, we can rejuvenate the body. So there's quite a lot of evidence now that that is the case. How much autophagy, this degradation mechanism, has got to do with senescence and these cells that should be eliminated and are usually dying is not quite known that, but it's something that we are trying to address, how autophagy relates to cell biology of senescence. We don't know yet. And which cells, senescent cells, are eliminated and which are not, particularly in the immune system, it's not very well known, but we know that the immune system plays a big role in eliminating senescent cells. And macrophages are, of course, their first phagocytosing, eating up these cells. And why we know that phagocytosis doesn't work so well with age, and that will contribute to the accumulation of these senescent cells, but we don't really know how they recognize and how it works. Sorry.
Thank you. We've got an audience question too. Spermidine enhances non-selective macroautophagy to suppress the aging of immune cells. Are there any specific cargoes, such as damaged mitochondria, that might be important in this?
So that is definitely the case, that if we knock out autophagy, we find more damaged mitochondria in the immune cells and in many other cell types of the body. We are currently setting up a system where we can label the cargo of the autophagosomes, and we'll let you know whether there's anything of any mitochondria in there, particularly in young versus old cells. But I don't have the answer. I think that mitochondria play an important role, and if you don't degrade your mitochondria properly, then you change the metabolism of the cell, and that will make a big difference to many things, function and differentiation.
There's another question that's just come in, brilliant as ever. That's the comment to you from the questioner. As well as the indirect effect via TFEB, is there a direct effect of spermidine? For example, does lysosomal pH decrease on spermidine treatment?
Yeah, we tried to look at that, Lynn. We couldn't really see anything, even though TFEB, of course, improves lysosomal biogenesis and is important for that. We couldn't really see an effect on the pH. I mean, we should have done, but we looked quite carefully and couldn't see that. So unfortunately, I can't say yes to that.
No. Well, I think I'll probably close this session because we're getting very close to the witching hour. I pass over to Dr. Paul Chen. I just have a comment on nutrition. I now begin to understand why Japanese and other East Asian diets are so good. I mean, I will never again disparage the idea of having terrific tofu in Kyoto or bean paste in Korean food, or whatever other various mixtures we are presented with. But this does emphasize a very important point. The diet is extremely important here, isn't it? Well, that's really, I suppose, a closing question to you, Katja.
Yeah, sure. I mean, fermented food is very good for you. It's got lots of spermidine, but we know that in general, fermented foods is excellent. Yeah.
Yes.
It's quite a difficult question to answer, what food do we eat and what is it, other confounding factors. So I think that is definitely not easy to understand. What else the Japanese do well that we don't do?
Well, there has to be some reason they've got so many centenarians. One final question. Would it be worth exploring something to increase internal spermidine rather than, as I take it, rather than actually consuming it as a product?
Yeah, I mean, we think that the most of the spermidine is either made by the cell or actually made by our bacteria in our gut. So changing the composition of the bacteria in our gut is maybe also what some of the right food does. So that's the internal sources. And, we don't know enough about that yet to really understand how we could use that. Probiotic or prebiotics might be a good idea.
Well, in closing this part of the meeting then, just once again, a big thank you to Katja Simon. We're privileged yet again in hearing from what I can call the horse's mouth, I suppose, of what's happening at the very front end of aging research. And with that, I hand over to Dr. Paul Chen, the co-host of the meeting, co-moderator, and I think he's got some news on a possible break. Over to you, Paul.